What's up, everyone? Today we are going to be going over developmental networks and how the expression of certain genes can either promote or inhibit other genes in a certain sequence, which can change the phenotype of individuals. So the way we're going to be going over this is using a fake sequence called spike, and that's going to cause spikes to grow on the skin of dragons. That's going to be our animal that we're going over. So, our example. So the way that this sequence is, you can follow it, is pretty self-explanatory. The S gene activates the P gene. If P is expressed, it prevents I from being expressed. You can see these little dead end and arrows either activate A or inhibit I. If I is not expressed, it can't prevent K from being expressed. If K therefore is expressed, it allows for E to be activated, and will cause spikes to be grown on the skin. This is the wild type, so this is the natural dragon phenotype. If we don't have P, then we can't prevent I from being expressed or activated. If I is activated, it prevents K, it inhibits K from being expressed, and then K cannot express E, and E therefore can't express spikes. If there's no I, then we cannot prevent K, and we therefore can express E, and there will be spikes. If there's no I or E, we don't have to look further than E, because obviously we cannot produce spikes. If there's no E, it activates spikes on the skin, so that's no spikes. And if there's no S or K, once again, we can go all the way down to the furthest spot and look. If there's no K, then it can't activate E, and if there's no E, then we can't have spikes on the skin. So that's how you follow a developmental network. Relatively self-explanatory. The way that we could have differences, even on cells that are right next to each other on the same organism, is through variations in gene regulation and expression, and a possible example of this or means of it could be transcriptional control. So when the gene is actually being turned into RNA, so like messenger RNA from DNA, for example, as you can see here, a type of transcriptional control could be a regulatory promoter that actually promotes the attachment and binding of the core promoter, which is the promoter that's actually going to express the gene in the end. Okay, thank you, bye.